to Jessica, who's going to tell us how to find that balance, because you're working on this. You've developed the Future of Research Symposia. You're talking about how to teach graduate students and postdocs to have a more successful future, bridging these types of gaps, addressing these questions. So I was hoping, Jessica, you could tell us a little bit more about the Future of Research Symposia. You've had 2 now, I believe.
2 in Boston.
2 in Boston. And so we'd love to hear about that and your ideas about addressing some of these issues.
Well, thank you so much, Rudy. I mean, part of the motivation for the Future of Research Symposia, which are really these grassroots organized meetings to bring junior scientists, early career researchers, together with other stakeholders in the research enterprise to try to understand how we can produce more efficient science. And so I'm so grateful to you for inviting some early career representation here today. We, as early career scientists, postdocs, grad students, are of course incredibly troubled by these very issues that I'm hearing now. The conflict between what we feel that we need to do in order to keep doing what we love and keep trying to promote science. And also the need to make that science all the things that we want it to be, that is creative, collaborative, open, and for the public good. The fact of the matter is that the environment that we're in is extremely competitive. And there's 2 aspects to that. First of all, about half of grad students entering their graduate school career, this is from research by Henry Sauerman, Michael Roach, have the intention of continuing in academic research, yet less than 10% of them are going to become tenure-track faculty. And so this disconnect between the intention of students entering and the reality, that's not to say that their PhDs can't provide incredible benefit, in industry, and indeed we should be thinking about going into those realms. However, we aren't necessarily always given the opportunity to explore and develop the skills that we need to apply our training outside. And whether that is through industry, through partnerships with industry, or through developing skills like entrepreneurship or communication. There's a bit of, I think, a conflict where in graduate school and postdoc, so often trainees are doing just research. And so this is a really great way of training researchers, but it may not be equipping students and postdocs with the skills that they're going to need in the broader world. And the second side of this coin is really this issue of how driven people are to continue in discovery. And we have this sort of exponential growth model in biomedical research where we have a PI training many students and many postdocs. And this is really not, I think, sustainable, especially during a period now where NIH funding is not growing. And so this leads to behaviors that are not productive for science. This is, I think, hurting science by the tendency for people to try to get that high impact paper, by concealing data, not being open about their findings, not sharing, not collaborating. And these are all things that I think every idealistic student going into science, from wherever they come from, wants to do. And I have known so many brilliant people who have chosen to leave because they find that the reality of research is not this dream. And so we, as a group of postdocs in Boston, together with groups in Chicago and the Bay Area and in New York, want to figure out some way. We would love to change this and love to make research better and more efficient for everyone.
Well, that's, you talk about the high impact paper, and I know so many of my own trainees, I'm telling them, publish your data. You don't have to shoot for Nature or Cell.
Yeah.
But about half of them would rather wait, 3 to 5 years going for that Cell paper or Nature paper, and very often, just because of the way the papers get reviewed, you don't get it in Cell or Nature,
Yep.
and you still publish it in a high impact journal, but you've spent 5 years without a publication. Finding that balance, do we need ways to better teach students and postdocs about finding balance of publishing medium impact papers while saving it up for the high impact paper so that they share their data more readily? Do you talk about that in your symposium?
Yes, I think the issue of publication is really important. And of course, publication, as you're discussing now, is kind of this primary currency for people who want to maintain a career in science, right? And so I think that the problem is that even though there are so many of us, myself included, who would love to just throw all of our data on the web, freely open, without any hindrances, this is not, we feel that we must play by rules that exist, rules that we don't like, rules that we would like to change, but things that we are kind of bound by. And so it's very challenging as an individual actor to make changes, especially when that scientist is in a vulnerable position. And I think it becomes even more challenging, if you're coming from an institution that is not, so fancy, or if you have a background that is otherwise disadvantaged. And so I think these forces are really pushing out a large fraction of our talent pool. And I would love to change it, but I think the root of the problem is the intense competition and the number of, the structure of the workforce, driving people to look for any way of gaining an advantage over others instead of working together.
Right. Well, we'll get back to this. So, Keith, you, as Vice Chancellor of Research at UCSF, I know you spend a lot of time thinking exactly about these issues, about how to train your graduate students, your postdocs for research in this century, dealing with this hyper-competitive environment we're in, measuring team science versus individual science. Would be wonderful if you could share with us your views of what do you tell your own graduate students and postdocs at UCSF? What's the advice you give them?
Great. So first of all, I should say that Jessica's going to come after me because I've trained 100 students and postdocs in my lab. Sorry about that. But I think that these issues of sort of forces of policy and tradition, especially within academia, actually, are things that really need to be addressed directly. And I hope that as this session proceeds that we'll actually dig into some of those in some detail. What I'd like to start with is thinking about the topic of redefining discovery in the 21st century and thinking about the scientific drivers that I think should really promote institutions kind of taking a step back and asking themselves, what are we doing? What are we actually training our students and postdocs to do? What do they need to have in their pocket to succeed in the 21st century biomedical research endeavor? And I would wager that if we did that, it would be sort of an embarrassing exercise, and that we would discover that we remained stuck in a training mode that is no longer relevant for research today or certainly tomorrow, and that in allowing that to happen, we've extended the training periods in ways that are costly not only to us as investigators but very costly to the trainees. We're burning up their most creative periods at a time when keeping them in training when they could be out working independently and so forth. So I hope we can dig further into that. But let me just start by, let me just comment in this phase by talking about three changes, scientific changes, that I think should have us thinking hard about the way that ways that we train and the things that we should be talking to our trainees about. First is that biology and biomedical research has undergone, or in some places is undergoing, a very dramatic transition from being a descriptive observational endeavor, from which we've learned an enormous amount, I might add, to a quantitative one. And there are lots of scientists from my generation, I would venture, that are doing biology because they wanted to do science but didn't want to do math. And that we're in a different world now. And so some of the changes that we need to make at the scientific level are a recognition of that, and that we need to have all of our trainees capable of understanding statistics, of being able to ask and answer questions with computers, being able to look at systems-wide problems and drilling all the way down to reductionist science in quantitative fashion. The second has been, as you mentioned in the introduction, Rudy, and that is teams. That is that the best science going forward is going to be done not by individuals, but by teams of scientists who actually bring different expertise to the table. This has lots of implications, but let me just mention one, and that is that traditionally, if your name appeared on a paper, you should have been responsible for every single piece of data in that paper. And actually, we've passed that era, interestingly enough, so that we will increasingly be involved in teams of researchers who know very different things, and that there will be authors on paper who don't know how to turn on the machine that gathers some of the data, or maybe even wouldn't even recognize the machine that gathered some of the data that are in the paper, right? So this means team building must be done carefully, thoughtfully, and with a lot of trust. And so there's this whole social dynamic aspect that's going to go into doing the best science going forward. And so team building is something that I think is going to be really essential. And then the last scientific piece is also a big change. And we used to train our students to design experiments in which there's only one variable, and it's the one you're testing, right? And that era has also gone by. And so I'll just lump this under the notion of robustness. And we are now, science is advanced. We should be celebrating this. The science has advanced to the point that we can ask and answer questions in systems and experimental approaches that are vastly more complex than having one variable that's under investigation. This has exploded recently in this whole question of reproducibility, right? But in fact, what it really means, so I characterize this as the Donald Rumsfeld problem, which is that there are unknown unknowns, that are a vast number of them, that are embedded in every experiment we do in these complicated systems. And we need first to acknowledge that and be thinking about how to move forward in our endeavor, knowing that there's lots of things that we're not testing in our systems. No reviewer would read a paper and send back a review that said, have you tried doing these experiments at 1/10 of a degree warmer? Or starting your experiments at 2:30 in the afternoon instead of 9 in the morning? Right. But there are variables embedded in our systems which those could be profoundly important. So those three scientific changes, I think, should have us stepping back and driving some changes in the ways that we train our scientists going forward, because there are things that they need to be able to become very facile with and accommodate the ways that they do their studies and the ways they talk about them to others that have big implications for being able to move science forward.
Thank you. You bring out a great point, which is building out the team of different disciplines and different investigators, but also in attacking the problem, bringing in teams of different proteins and genes and networks and broadening out from single molecules to systems, broadening out from single investigators and single disciplines to multidisciplinary approaches. I think that's a great theme that we can continue on as we talk more. And as you do things like this, as you start broadening into giant systems, it is so important at that point to bring in your other 2 points, a quantitative approach, good statistics, and robustness, because it's very easy to go awry when you're trying to, if your model becomes too heuristic, it's very easy to lose the relevance back to the initial question, or especially to translating that to helping humankind with therapies. So we'll definitely come back to that. Thank you. So last but not least, Murray, bringing us the pharma view. You've been very actively thinking about how can we better bridge what's done in pharma and at the clinical end with basic sciences. We'd love to get your thoughts on that. And also, are we focusing too much on disease? And should we be focusing more to create that bridge on pathways and platforms as opposed to just individual diseases as we think about pharma academic collaborations?
Great, thank you very much.
Yes.
Well, I'm delighted to be here to work with Faster Cures, because one of my motivations is getting our medicines to patients faster than we currently do. Currently the timelines really aren't great. It takes 10 years to develop a medicine. It costs over 2.6 billion. And clearly, for the sake of patients, we need to get better at doing this. One theme that's clearly come out today is collaboration. So pharma can't do it on its own. In fact, it doesn't want to do it on its own. Probably half the medicines that GSK develop are internal, but the other half are to do with partnerships. And partnerships, to me, is vital. And I think it comes back to the point about what you bring to the table. And we can bring lots of things to the table in terms of understanding chemistry, understanding translation. And I want two areas to talk about collaboration. One is we do a lot of work with institutes like the Francis Crick Institutes, where they can bring ideas around genomics. We can bring various hits and targets. We work with the European Bioinformatics Institute. So we do work with big institutes, and the collaboration is great because you put all ideas on the table. Sometimes it's best to forget your background and just come to the table and to discuss ideas. But I want to pick up something that Keith said, because pharma is interested in any ideas and lots of ideas, and it's also interested in working with PIs that may be struggling. And what we've recently set up is what we call academic discovery performance units. So within our own company, we've got little discovery performance units, and some of them have 10 people, some have 40 people. And we certainly know once you've got a critical mass, you want people to work together. But we've noticed that a lot of academics have got a good idea, and they may understand the biology, but they don't understand the chemistry. So they don't know how to make a molecule. They don't understand finances. So how do they go from their biological idea to develop a medicine? Now, what I think's happened in the past, people have been going around looking for a great idea, spend a large amount of money, but not think of the gap in between. And I think one way to solve the gap in between is start doing it in small steps. So with some of these individual PIs, we've set up a risk and reward sharing. So you go by various milestones. So what's your hypothesis? My hypothesis might be that I think this is an important target. Okay, next stage, can you actually make a molecule? Can you actually get the chemistry right? Okay, once you've got the chemistry, how do you test it preclinically? So this may seem obvious, but I think it's important to go together, sharing the risk, sharing the reward, going by milestones. And that's been far more productive than going around looking for the needle in the haystack. And coming back to a little bit about the translation science into medicines, I want to give an example a lot of examples are used in oncology, but I want to give an example in respiratory medicine, where you've got to be more precise and you've got to understand the biology. And I want to take interleukin-5 and an antibody we've got against that. So we know that by targeting that, we will lower eosinophils. And you think, well, lowering eosinophils may be important in asthma. And the first study we did with IL-5, we took people with mild and moderate asthma. We looked for the standard endpoint of FEV1 suggesting improved lung function. No effect. We nearly walked away from this, apart from some bright scientists in the lab said, well, I've looked at the data, and I know I'm not really encouraged to do post-hoc analysis, but I did, and I found out the people who did best with IL-5 were the people who had really high eosinophil levels. And maybe we're going too broad. And I think one mistake big pharma has done in the past is it's tried to go for big wins. Or let's find the answer to asthma or Alzheimer's. And I think it's better to be more specific. So with this example, we said, well, okay, it does seem to work in people with high eosinophils. And it may be an inflammatory component. So instead of looking at the classical endpoint of FEV1, let's look for exacerbations. And let's only give this drug to people with severe asthma with high eosinophils. Now, some of our marketing colleagues say, well, that's no good. You're going narrow and narrow. And I said, no, the science is driving us there. And if we go there, we will get the most clinical benefit for the patients. So we deliberately targeted people with severe asthma who are not controlled on high-dose severe steroids, who are going into hospital with exacerbations and really have high morbidity and high mortality. By targeting those patients, we actually showed a very good response in the severe end, reducing exacerbations, reducing morbidity and mortality. So by being specific to the target, we're able to help specific people improve their quality of life. So this is where I think, rather than thinking broad, being more specific has clear benefits. But I really want to end is, I think what, Shiva, what you started with and set the tone is, we should not be biased about perceptions. People have myths and ideas. I think no matter whether you come from pharma, a small science background, what age you are, if you're motivated and have good ideas, we need to find ways of translating that idea into medicine.